Greetings. So we're going to talk now about the chapter in your book on DNA. So we're jumping ahead a bit. So if you are unfamiliar with atoms and covalent bonds and hydrogen bonds, review those early chapters before going full on into this one, just to refresh your memory on those topics. I will bring them up as we go through the chapter, but they are very important. Okay, so in the 1900s, the early to mid 1900s, scientists were trying to figure out what the molecule was that was passed on from one generation to the next, that allowed for evolution to occur, that allowed for inheritance to occur, that caused children to look like their parents and things like that. Gregor Mendel, whom I'm sure you remember, he did Punnett squares, we'll talk about him more later, figured out some patterns of inheritance, and when his work was rediscovered in the 1900s, scientists started to ask, well, what is the molecule that's passed on? And we now know it's DNA, of course, and the central dogma of molecular biology says that DNA codes for RNA, which codes for protein, and I know you've heard that before. So DNA codes for RNA, and the process of going from DNA and RNA is known as transcription. This will be in the next chapter. RNA to protein is called translation. This is basically going from the genotype, which is the information in your genes, to phenotype, which is the physical expression of genes. So eye color, hair color, whether or not you can digest milk, all of those sorts of things are a phenotype, the expression of the genes. Now, Gregor Mendel knew that there were factors passed on from one generation to the next. He talked about, big G, little g, or big P, little p, those sorts of things, and that these were somehow in the cell, but he didn't know much about it. Later on, we figured out that those factors were actually on chromosomes. So here's a chromosome, and that there were these areas, like where a big P might be, like let's say purple flower color. There are these areas that we know we called genes, and we know they're part of chromosomes. And when you examine a chromosome, it's made up of DNA and protein. So it's DNA wrapped around protein. And so one of those two things has to be the molecule of inheritance. Okay? One of those two things must be that molecule. So the question was now, which one is it? And scientists were pretty sure it was protein, because protein is really complicated. It's heavily folded up, lots of structure in a protein. Okay, so proteins are very complicated molecules. And so they thought, okay, this must be the molecule that is passed on from one generation to the next that causes all the traits of a human, for example, to be passed on from one generation to the next. Whereas DNA seems really simple. It just has a couple parts, A, T, C, and G. It's got 4 basic parts that we'll talk about in more detail later, but 4 parts that aren't very complicated, and it doesn't seem to be complicated enough to code for a human, a giraffe, an elephant, a bacteria, and a squid, right? That doesn't seem possible. So they assumed it was protein, but you can't assume in science. You have to test to verify, right? So Griffith in 1928 does a basic experiment to try to figure this out. First of all, he takes this bacteria called Streptococcus pneumoniae. It's a bacterium that causes pneumonia, and it comes in two kinds in this species. One is called the S type, and the other is the R type. The S type looks smooth under a microscope because it has, as you can see in this drawing, has like a membrane around the bacteria, and so they look shiny and smooth. The R type does not, so it looks rough. Now, the important thing to note here is in the control experiment, which we see on the left, if you inject a mouse with the S type, it gets pneumonia and dies. If you inject a mouse with the R type, it does not get pneumonia. So they say the S type is virulent and the R type is non-virulent, meaning it does not cause disease. Now, here's when the experiment gets interesting. He kills the S bacteria with heat. He knocks it out. And then he injects it into a mouse, and the mouse is fine. Okay, now the next part. He takes the dead S cells, and he mixes them with living R cells. So S cells would cause disease, but they're dead, so they can't cause disease. And he mixed them with living R cells, which do not cause disease, and he puts them together, injects the mouse, and the mouse is dead. He takes a sample from the mouse, and he finds living S cells inside. So somehow, living R cells were converted to living S cells. And so he calls this process transformation. Somehow, the phenotype of the organism was changed. It was transformed from one type to another. We went from R to S by mixing R cells with dead S cells. What we now know is DNA leaked out of the dead S cells, got into the R cells, and caused them to become living S cells. But he was trying to figure that out, right? Now, that was cool. He still doesn't know if it's protein or DNA, but we have something to work with now. So later on, as we know, science progresses and we build off the work of others. Oswald Avery comes along and some others as well. And what they do is they play around with enzymes that dissolve protein or dissolve DNA to see what happens. And they figure out that it was DNA that caused the transformation. But science isn't ready to fully embrace that yet. People are really sure it's protein. It's got to be protein. Proteins are so complicated. But Avery says, no, it's not. But we have to have further tests. And that's when we get to the next stage, which I'll talk about in the next video, the experiment with Hershey and Chase.